Good day and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. Our speaker today is Dr. Lucina Uddin. She is Associate Professor at the University of Miami, where she's also the Director of the Cognitive and Behavioral Neuroscience Division. In 2001, she completed her Bachelor of Science degree in Neuroscience and Philosophy at the University of California, Los Angeles, and completing her Ph.D. work in cognitive and psychological neuroscience at the University of California, Los Angeles in 2006. Her professional experience includes postdoctoral fellowships at New York University and Stanford University, and since 2014 has been a faculty member at the University of Miami. At Miami, she leads the Brain Connectivity and Cognition Laboratory, which employs neuroimaging and accompanying methods to examine the relationship between brain connectivity and cognition in typical and atypical development. Many of her long list of accomplishments includes the University of Miami Gabelli Senior Scholar Award, the Universal Scientific Education and Research Network Laureate in Medical Sciences, the Organization for Human Brain Mapping's Young Investigator Award, and several others. Her lecture today is entitled Neuroinformatics and Cognitive Ontologies, and we are very excited to welcome her to our series. Now, before we begin, as always, we are streaming this lecture live and for recording via YouTube. If you are watching on YouTube, thank you for joining us. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Uddin via the chat feature of their Zoom sessions. I'll synthesize these questions and ask them on your behalf during the last 10 minutes or so of Dr. Uddin's talk. With that, welcome, Lucina. We are very much looking forward to your lecture.
Thanks very much, Dr. Van Horn. It's really a pleasure to be here virtually with all of you, share some of our work in this area. And I do wish I could be there in person for all of these different activities, but until then, the virtual format will have to do. So what I'd like to share with you today is just some thoughts and some data about the topic of neuroinformatics and cognitive ontologies. So for those of us working in cognitive neuroscience, we know that there have been decades and decades of fMRI, functional magnetic resonance imaging work, that's identified distinct patterns of brain activation that occur while participants perform different kinds of cognitive tasks. And the goal, of course, is to map the areas of the brain that are involved in different types of cognitive processes. It's sort of the goal of cognitive neuroscience very broadly. And what we'd like to suggest here today is that these data can, in principle, be used to provide the basis for constructing a biologically informed, data-driven ontology or taxonomy of psychological processes. And there's many others who've worked in this area too over the years, and I'll draw on some of that literature as well. So in this talk, I'd like to highlight some of the progress and challenges associated with the construction of cognitive ontologies using informatics approaches and based on functional neuroimaging data. So let me go the right direction. Let's see. Okay. So what is a cognitive ontology? So an ontology generally is a branch of philosophy that deals with the nature and organization of reality. So what things are there in the world out there? How do we carve up nature at its joints? That's really what ontology deals with. And so a cognitive ontology then would be a formal description of concepts that are assumed to exist within a particular domain. Here we're talking about psychology and the interrelationships between them. So one sort of easy way of thinking about ontologies is just what are the knowledge structures that we know about that we use to communicate amongst each other. So, we just saw the NBA finals with the Lakers coming out as champs again. And if you think about an ontology of the NBA, you have things like basic units like players that belong to teams, and you have a Western Conference and an Eastern Conference. And knowing this knowledge structure tells you that in the finals, you're going to have a team from the West play a team from the East, because that's a structure of the NBA. And knowing all the things we do about teams and their compositions and the players that make them up, it tells us something about what we can expect in any given category to contain. So just knowledge of the way the NBA works is a particular ontology. And just like that, we we had these sort of knowledge structures in various domains. So, in the case of cognitive ontology, we might think about something like a particular task and how we should categorize that. So many people use these simple task paradigms. The flanker task is one in which the participant is asked, look at the arrow in the center and tell me if it's pointing to the left or pointing to the right. And you use the button press to indicate your response. And the challenge of this task is sometimes there's flankers to that central arrow going in opposite direction. And anytime you do this behavioral paradigm, it slows people down when there are arrows pointing in the opposite direction of the center arrow. This task has been around for a long time, and we use it, in the laboratory. And the question is, what are we using this task for? What cognitive process do we think we're tapping when we do a task like this? And a cognitive ontology, the research in this area would actually formalize this kind of question. So if you had, for example, so if you had one individual who says, well, this is a response inhibition task, that's what we're measuring here, the cognitive construct of response inhibition. And then you might have another scientist say, no, what we're really looking at when we ask people to do this is error detection. That's what I think the flanker task is mapping. And in what we do when we look at cognitive ontology research is formalize this type of disagreement into a research program where we can say, well, let's figure out what cognitive construct or process is this task really tapping, and how do we get to the answer to that question? What is a way to sort of create a better cognitive ontology or categorization of these mental and psychological constructs? So there's several approaches for constructing cognitive ontologies, and they can sort of broadly fall into the category of bottom up and top down. So a top-down approach is where you would use expert knowledge and opinion to get a group of researchers to agree, well, when you do this task, what you're really engaging is this cognitive construct, and now we all agree on that, and we can write it down and use it to communicate. So there's been a lot of work in this area using expert opinion and topic modeling of texts in article databases. You see some of the references here: Russ Poldrack's group, Angie Laird, Jessica Turner, Talia Arconi have done a lot of this work at building a top-down sort of cognitive ontology. And of course, the benefit to something like that is that you can, produce a detailed ontology of relationships and entities. But there is, of course, the downside that this may fail to map onto actual divisions or categories of nature. And there's some other downsides that I'll mention in a minute. But this has been one sort of popular approach for construction of a cognitive ontology. And on the other hand, you have this bottom-up approach, which is usually a data-driven or more informatics-based approach. So data-driven classification or clustering of functional imaging or behavioral data. And you can also use supervised machine learning for this type of thing, or exploratory unsupervised machine learning. This bottom-up approach basically, can try to derive categories of cognitive processes directly from task fMRI patterns. And the pros of this is that an ontology derived in this manner might more likely reflect actual divisions or categories of nature that we see, but they may not necessarily provide enough detail to be of practical use for knowledge representation. So, every sort of approach has its pros and cons. So one example, just to, that many people might be familiar with, is of a top-down approach, is this research domain criteria that the National Institute of Health has sort of put forth. And this is a way, it's a sort of a call for new ways of classifying mental illness that are based on dimensions of observable behavior and neurobiological measures and other measures. And this is just a real kind of summary of some of the categories within the research domain criteria. Back in 2016, I was part of this cognitive group, the blue group here in the middle, that basically it was a group of experts who decided what the categories were that would be of use for study, to standardize across psychiatric disorders and to push forward personalized medicine and so forth. And being on the panel of, on this top-down panel made me realize sort of firsthand what were some of the downsides of doing the top-down approach. And one of them is, of course, everyone who studies cognitive neuroscience has their own biases and their own sort of lifetime of ways of thinking about things. And so expert-based approaches are prone to the kinds of biases that experts have. So you may be a person who studies executive function, so you think these particular aspects of cognition are the most important or the most salient or the most interesting for investigation. Doesn't necessarily mean that's the way the brain is carved up or that's the way that the mind works. But so, that this was a top-down kind of ontological approach. But the reason that this sort of research domain criteria has been so popular is that the goal was to carve up psychiatric disorders in a way that's not just symptom-based. So it's not just you see a person with observable symptoms in this manner and you put them into major depression or bipolar or some other category. Because, of course, symptoms themselves cannot tell you much about the underlying mechanisms. So perhaps if you look at genetic risks, brain activity, physiology, behavior, and life experience, and derive data-driven clusters of individuals who were more similar within cluster than across cluster, those people might not all fall into the category of major depressive disorder, but they might all be most well treated by a particular type of drug or intervention, because once you've identified clusters that are more homogeneous, the treatment and kind of response research can be more targeted and hopefully more efficient. So RDoC has really prompted individuals in the field of psychiatry to rethink how psychopathology might be best served by building a framework that's based more on biological and cognitive validity. So, a lot of people working with the RDoC framework now look for research findings that cut across diagnostic boundaries. And, this is all towards the sort of goal of precision medicine. So that you can see why, despite there being flaws in the top-down approach, there's been a lot of success with this method. And there's been a lot of work, this is from Russ Poldrack and Talia Arconi, trying to derive things like cognitive atlases, which carve up the space of all the different cognitive constructs in a way that lets us use a common language to communicate about them. So this Cognitive Atlas project is basically a collaborative knowledge building project aimed at developing an ontology characterizing the current state of cognitive neuroscience. And so this nice paper summarizes some of the constructs you see all the time: cognitive control, working memory, executive function, and what tasks measure them, that tasks that we use as scientists, and sort of how the different things map onto each other based on basically what the field is already doing. So this is just a summary of what is actually going on in cognitive psychology and cognitive neuroscience and cognitive sciences more broadly. And as you can see, this, similar to what RDoC is trying to do, the goal here is that if you, understand a particular process like response inhibition, which we then map onto a brain region, which we think, well, ventrolateral prefrontal cortex seems to be involved. Then you can look at things like signaling pathways, genes that contribute to function of those regions. And then at the highest level, syndromes in which that particular function is disrupted and cutting across different diagnoses as well. So all of these projects kind of get at the same goal of relating biological functions and processes eventually to things like psychiatric syndromes. A similar effort is called the Cognitive Paradigm Ontology, put forth by Jessica Turner and Angela Laird. And this was based on a drawing from the taxonomy of the Brain Map project, which many of you are probably familiar with. The Brain Map database includes coordinates from, thousands and thousands of task activation studies that have been coded in particular ways. And what the cognitive paradigm ontology does is represent the characteristics of the cognitive paradigms that have been used in fMRI literature over the years. So this is the Brain Map database itself includes these categories like the experimental paradigm that was used and the type of stimulus, the response type, all these different sort of categories that are used to characterize the broad database of fMRI studies that are available today. So this is another sort of way of organizing what's out there. And this is just one example that I like. Why am I going backwards? There we go. Another example I like of trying to identify cognitive components from neuroimaging work from Thomas Yo's group. And here he basically used an author-topic hierarchical Bayesian modeling approach to apply this to contrast from the brain map database to derive what he's called here cognitive components that may contribute to performance of specific tasks like working memory or attention tasks. And a very nice sort of mapping between experiment, task, cognitive components, and the brain regions involved. This is a very interesting paper that I recommend for those who want more on this topic. The reason I particularly like this paper is because he quantified what brain regions use the most number of cognitive components. And really interestingly, there's the areas here in red you see, one of them is the anterior insula, is basically activated in many, many, many tasks and paradigms and experiments. And as you can see, it's got a very high number of cognitive components. A lot of my work in the lab I'm not going to be talking about today focuses on the role of the anterior insula and brain network dynamics. And I think it's just very interesting that you can use completely data-driven approaches to show the same thing that we've been doing using things like meta-analytic connectivity modeling and dynamic functional connectivity to look at the flexibility of the insular region. And here you can see the flexibility here is associated with the fact that this region just participates in all kinds of, all manner of tasks. So really interesting findings. And so what we, what I'm gearing up to tell you about today is some of the work that Taylor Bolt in the lab did when he was a PhD student. And he left the lab a few years ago and is now a data scientist at Deloitte, but he's still publishing with us, even though he's taken a position in industry. So really fantastic and interesting work for his dissertation here, where he began a project that was sort of a data mining cognitive ontology discovery project, looking at these task fMRI data repositories with the goal of mining them to look for clues of how patterns of bold activity might delineate categories of psychological function, following up on some of the, the work that I showed you, the introductory work that I showed you. And the goals of this project here that was done in collaboration with Angie Laird and Simon Eickhoff and others, I think this just came out in Neuroinformatics in 2020. The goal here was to identify data-driven dimensions of a potential ontology based upon the ability of that ontology to explain variability between patterns of brain activity. And the approach he took here was a whole brain representational similarity analysis to quantify spatial similarity among task activation maps within BrainMap. And here the assumption is that important features of existing ontological categories could be identified in terms of the amount of how much variance they explain in task activation map similarities. So that was the the goal in this project. So what he actually did in this set of analyses was applied an unsupervised clustering approach to all the task activation patterns that could be derived from the BrainMap database coordinates. And here this analysis included over 15,000 activation maps that yielded almost 9,000 contrasts. That's contrast between different conditions in an fMRI task. And they were all, of course, derived from activation peak coordinates that are what are available in the BrainMap database. And this involved a data-driven dimension reduction and a multivariate distance matrix regression. So, so basically, we started here with, this is an unsupervised clustering approach applied to the entire corpus of the reported activation coordinates in the BrainMap database. And he began here with the construction of activation maps from these reported peak coordinates of task fMRI studies. And then a dimension reduction step, reduction of these voxel level maps to a smaller set of networks using sparse non-negative matrix factorization. And then a clustering of basically an activation map by activation map similarity matrix based on the similarities in network and activation estimates between the different activation maps from these coordinates. So there were two goals. One was sort of to identify how closely the labels in the BrainMap database really explained differences in task activation patterns across this huge database. And the second goal was to see if we could use a more bottom-up approach to derive latent categories directly from the data. So the way this worked, well, interestingly, the unsupervised clustering of all these brain map activation maps yielded a 4-cluster solution that achieved the most replicability using a cross-validation approach. And this 4-cluster solution could be sort of divided up into these broad categories of tasks: object viewing, complex action, affect, introspection, social, and auditory motor. But note, of course, these sort of 4 clusters showed a lot of overlap in the activation maps that were associated with them. So, the only cluster that didn't look a lot like the others was the affect social cluster, which resembled a little bit the default mode network, which we'll talk about in a bit. But what this type of analysis suggests that the current, so I forgot to mention that the current labels in the brain map database didn't explain as much of the variance in the task activation map as these latent clusters. So this suggests that the current descriptions of cognitive processes and the task used to elicit them do not necessarily reflect activation patterns commonly observed in the human brain. And this is just to note that the 4 data-driven latent dimensions explained a greater variation among activation maps than all the conventional labels that were already present in the brain map database that were used to categorize the tasks in the brain map database. And this is just showing that the sort of tasks, the default mode and the negatively correlated frontoparietal systems is sort of a ubiquitous pattern that you see in resting state fMRI as well. We'll get back to that a little bit later in the talk. And this is just a little bit more of a view of the 4 activation profiles and the associated constructs with each of them. And this will be probably not so easy to read here, but you can find the full table in the paper if you're interested. It's just another view of the cluster 1, 2, 3, and 4 in terms of the different task paradigms and different constructs that seem to be associated with these types of activation maps. And this was just on the left showing you how we determined the number of clusters. And there was both a 4 and a 7 cluster solution that showed high levels of stability. But if you look here on the right, the 4 cluster solution and the 7 cluster solution derived in a data-driven manner really explained more variance than the traditional brain map labels on the bottom, which are behavioral domain and task paradigm, for example. So basically the similarity matrices here with brighter colors within the red lines and cooler colors outside are showing you that there's sort of more well-separated groups of task activation maps in the top 2 matrices than in the bottom 2, meaning that the cluster solution that he derived in a data-driven fashion was better able to explain differences in activation maps than the labels that have been provided already by the users in the brain map database. So in summary here, task paradigm, which is one of the categories that brain map database includes, seem to explain more variance among task activation patterns than other dimensions, including the latent categories that you saw there. And this means that like when an individual is deciding on particular task to study a particular construct, like working memory, for example, that which paradigm they choose can be very important because different task paradigm as a category explains a lot of the variance among these activation maps. Study ID, which is which study the activation maps came from, actually explains about 50% of variance in activation patterns. And the data-derived latent activation states we saw here were associated with kind of known functional systems, frontoparietal, salient, sensory motor, and default activation patterns. And those 4 data-driven latent dimensions appear to explain greater variance among activation maps than all the conventional ontological dimensions combined. So these are just some clues that just because we think, well, we have a working memory task, or we call it a working memory task, it doesn't necessarily share activation patterns with other things that are called working memory tasks. And in fact, the labels that we've been using all along, may need to be revisited if we're really looking for things that explain great variance amongst brain activation patterns, then the current categories that are used, the current labels that are used, don't seem to be doing a great job of that. So that was, there's another kind of way to ask this question. The first analysis was trying to classify activation maps into separable categories and have separable clusters, but not sort of account for commonalities between them. So there was a lot of overlap in the clusters I just showed you. However, that's because the goal of that unsupervised clustering algorithm was to highlight the differences among the maps, not the commonalities. So the next goal was to sort of explore the commonalities that were observed in an exploratory model. And here, what he chose to do was a bifactor analysis to explicitly model a general commonality that could exist across all activation maps while allowing for subclusters to emerge within this general pattern. And this analysis, so this analysis required us to accurately estimate similarities and differences between activation maps. And the peak coordinate meta-analysis, that is permitted by BrainMap wasn't quite sufficient for this job. What we really wanted here was unthresholded activation maps. And we found those available in the NeuroVault database, as well as the Human Connectome Project. Those are two places where unthresholded whole brain activation maps are available. So, using all the task fMRI activation maps available in NeuroVault and in the Human Connectome Project yielded 108 activation maps across a variety of task domains that we could use in order to follow up or to ask this question. So, so the approach here was to first do a data reduction to reduce the spatial dependence between the activation estimates across voxels. Then we conducted an initial extraction phase in which an initial factor solution is estimated and the appropriate number of factors is determined by parallel analysis, followed by a bifactor analysis phase in which the factor analysis is re-estimated with the estimated number of factors from the initial extraction phase and a bifactor rotation criteria. And finally, testing the fit of other alternative factor structures using confirmatory factor analysis. So how did this work? So this is just a task activation conjunction and task deactivation conjunction across all those maps. And as sort of a sanity check, you can see a lot of frontoparietal and insular and cingulate systems are activated across all tasks, and deactivated default mode network regions, as one might sort of expect. So, so basically, the first step was a data reduction, as I mentioned, and this was sort of followed up by the bifactor analysis approach, where each task activation map representing activation here in red associated with a particular task is modeled as arising from direct effects of a single latent general factor and various latent subfactors. So this was a way of sort of first accounting for all the commonalities we see across task activation maps, regardless of task, as well as sort of activation or sub factors that account for differences among different task activation patterns. So this results of this bifactor analysis suggest a general factor which explained over 50% of the variance in the activation maps. And again, it looks very familiar to sort of task positive, task negative, or default mode and frontoparietal systems that we see sort of across all kinds of both task and rest paradigms. But within the, in addition to this general factor, we saw as well subtypes, which explained an additional 31% of the variance in activation maps. This suggests that that there is sort of a hierarchical structure to these patterns that we have observed. So what else do we know about this pattern of the sort of midline regions with the lateral frontoparietal regions? So this kind of lateral frontoparietal system is independently described as a multiple domain system or domain general task system or the extrinsic mode or the executive control system. It just goes by a lot of names. And I'll bring up this point again in a few slides about what I think about the problem of all this nomenclature. And then there's the sort of midline regions that are often described as a default mode network. And of course, there's seems to be antagonistic relationships between these sets of systems. And the pattern that you see is pretty ubiquitous if you look at gradients of functional connectivity here on the left with Margulies, if you look at coactivation pattern analysis, if you look at the posterior cingulate and its coactivation, again, you get this default mode network. If you look at correlations of brain areas with amplitude of EEG alpha rhythm, this pattern of sort of lateral frontal parietal versus medial frontal parietal regions is ubiquitous across different analysis techniques, research questions, task or resting state fMRI. So this shows that the single activation pattern dominating most observed task fMRI activation maps is this positive negative pattern we've seen over and over again. And any cognitive ontology derived from an exploratory analysis of task fMRI activation map seems to be, it should be dominated by this pattern. So understanding what this pattern represents is, I think, essential for any cognitive ontology or any construction of a set of basic principles by which cognition is organized in terms of underlying brain activation. And that all of this brings me to our sort of current work, which centers more around brain networks in cognitive neuroscience. So I've been talking a lot about individual brain areas that are activated or not in response to specific cognitive tasks or specific instructions or specific paradigms. But what our lab really focuses on a lot these days is network approaches and network neuroscience, and understanding that brain regions don't really work in isolation, right? And you have sort of patterns of frequent co-activation and patterns of functional connectivity that are sort of underlying a lot of what you see when you look at task fMRI data. And one thing we've sort of been trying to tackle in our recent work is this problem that people refer to different brain regions belonging to different brain networks in a way that's not at all standardized. For example, for a region like the anterior insula, you can look at the cognitive neuroscience literature and find people saying, well, the anterior insula is part of the singular percular network, the anterior insula is part of the Stanley's network, the anterior insula is part of a ventral attention network. And they may also ascribe overlapping yet unique roles for these brain regions within these networks. So, I see this as a problem for the field in terms of developing a common language, developing taxonomies that we can use to communicate amongst ourselves. So how are we going to define a functional brain network given we don't have this consensus at present? And how many functional brain networks are there? These are, of course, ill-posed questions, but they're sort of what's keeping us up at night, right? So, I started to do this work with Thomas Yeo and Nathan Spring last year, where we tried to move towards developing a universal taxonomy of macroscale functional human brain networks. And I think this just relates a lot to the work that's being done in the construction of cognitive ontologies, I think for obvious reasons. And this was an initial stab at this difficult question that we took. And this was just the stance that if we start to use similar terminology across studies, we may really make progress in terms of human brain mapping. And if we don't have conventions and we don't have consistent nomenclature, it will just hinder future progress in this field. So luckily, I was able to convince the new OHBM Best Practices Committee that what we really need is to have a committee of individuals working on this topic who have worked in connectivity, who have worked in meta-analysis and who've worked in anatomy, to come together and see if we can reach a consensus of sorts on this really important nomenclature issue. So the first thing we did initially was just look at what's already out there, and almost every independent component analysis or brain parcellation or connectivity modeling approach, meta-analytic approach, almost everything identifies something with a visual back of the brain network, as you can see. And here is Steve Smith work on the left comparing resting state networks to the brain map database meta-analyses. On the right, Thomas Yeo's well-known functional parcellation in the brain. On the bottom, one by Evan Gordon. And you can see there's always seems to be something called a visual network or an occipital network. So maybe that should be one of the things we include in our taxonomy of networks when we eventually build one. You can also almost always see something like a somatosensory or somatomotor or motor cortex network. You can see it in resting state parcellations here, on different resolutions. And if you obviously parcellate the brain into smaller and smaller bits, you can subdivide these networks into subsystems. So this seems to be a place we can start. And then you get into these higher level cognitive association areas. There's been what's been called the dorsal attention network from many years of cognitive neuroscience research showing that sort of top-down attention engages these frontal eye field and posterior parietal areas. And meta-analytic connectivity modeling suggests that something like this exists. And you can see in green, the dorsal attention network comes out in several different parcellations of the whole brain. So these are, we think, some starting points. This one, and as we get further along, we get into more controversial perhaps and less agreed upon nomenclatures. But people talk about executive control, central executive, multiple demand, as I mentioned. This network seems to have a lot of names. On the right, it's classified as frontoparietal, and on the left, these are from executive individuals who refer to these networks as executive control or central executive networks. So there just needs to be a little bit more agreement upon what label we should use to describe systems or activations or connectivity within these regions. And here's one that's close to my heart and also very controversial. In my work, I've called the insular ACC network the salience network. But there seems to be a lot of overlap anatomically with what's been called the ventral attention network, as well as the cingulopercular network. And there seems to be a lot more variability in terms of the spatial topography here. And, this one will be a difficult one to come to a consensus on. But the nomenclature that we're proposing here is more anatomical based and less and more agnostic with regards to what we think the actual function is. So if we see mid-singular insular, connectivity or activation, we might just say, okay, well, that's a mid-singular insular network. And that will prompt us to, more consistently identify this in future work. And this, of course, the default mode network, everybody has known about for many years. And we're even proposing to rename that as a medial frontoparietal network with the idea that sticking with the anatomical terms will help us moving forward. And of course, you see fractionation within this network. You see subsystems. It's not a sort of monolithic entity. So this is where a lot of work needs to be done to parse that and what we should call these subsystems, for example. So, this is one example of I would say this was a top-down approach at deriving a taxonomy of functional brain networks. This is the initial breakdown that we observed or that we agreed upon. But again, the reason we formed the current best practices committee was because we really need sort of wholesale buy-in from the community in order to make any taxonomy work. So it doesn't really make sense for me to call something a mid-singular insular network in a paper if no one else is adopting that terminology and no one else knows what I'm talking about. So these are some of the ongoing challenges and goals for our current best practice committee on network nomenclature. And why we still need to refine this taxonomy should be obvious. I gave a Twitter talk on this a few months back about what about network dynamics? What about inter-individual variability and activation and connectivity? What about subcortical brain regions? What about the idea that networks fractionate into subsystems and the hierarchies I just talked to you about? What about these hierarchies? These are all things that require a lot of discussion. And so the group we have assembled is now meeting once a month to hopefully tackle these issues and see where we get. And I think one of the things that will make this program or this goal hard to achieve is that networks don't just kind of stay in one place and do the same thing from moment to moment. There's really nice theoretical paper by Marcel Messelenbeck from the 90s talking about how cognition is subserved by interconnected neural networks and complex behavior is mapped at the level of multifocal neural systems rather than specific anatomical sites. So even back in the day before fMRI, we knew that it wasn't as simple as one brain region does one cognitive process. Multiple brain regions contribute to multiple processes. And you could see that even with a neuropsychological approach. We also know that the function of a brain region and brain area can often depend on the status of other connected areas. This was the idea of neural context put forth by Randy McIntosh. So a set of nodes here, for example, the gray nodes might behave in a particular way when the black nodes are there activated, but behave in a different way when the black nodes in B are activated. So this context is important for the function. And lastly, something we're very interested in is dynamics and how regions change, brain regions change their affiliation over time in the matter of seconds and probably even milliseconds. And so a network, of course, needs to be understood in terms of the interactions between multiple brain regions as they unfold temporally. So I think because of all of these issues with respect to dynamics and many-to-many mapping of brain region to function, all of these issues will make it really difficult to arrive at a taxonomy or ontology of brain networks, but nevertheless will persist. So this is just a short overview of how I think neuroinformatics can contribute to the formation of cognitive ontologies. This work was really done by Taylor Bolt, and congrats to him for pushing our lab into this direction. And Angie Laird has been a great collaborator, Simon Eickhoff, Thomas Yeo, and Nathan Spreng. And of course, the funding sources have made this all possible in many ways. So I wanted to just close by saying, vote like your life depends on it, because that's been the only thing on my mind for the last few weeks. And I just thought, if we haven't yet, please do. And this is our, the only hope that keeps us going at this point. And now I'm in San Diego on sabbatical, so this is my sort of morning view, and I'm happy to leave the rest of the time for questions, and want to say thanks again very much for this kind of invitation and for providing me the opportunity to share some of this work that I've been very excited about. I'm happy to take any questions now. I'll hand it back over to you, Jack.
Thank you, Lucina. That was a fantastic overview of cognitive ontologies, many of the challenges which exist. And it's just an area which I have observed, either been involved with those discussions or observed for many years. And you're absolutely right. It's a very challenging problem. And one of the things I think you kind of, I want to encourage our Data Science Innovation Lab participants to send in any questions via the chat. But while we're waiting for a few, I was you touched on a couple of key things. You have the kind of the expert opinion about how the ontology ought to be constructed. And I remember you mentioned Russ Poldrack a number of times, and he's a colleague and friend. And he has mentioned to me several times that trying to get a bunch of psychologists to agree on what to call things. You can maybe get them to agree on the topmost layer of the ontology, but you get about 2 layers in and it becomes a probabilistic fog of what people are going to call it. And so there's this sort of lack of consensus with regard to these kind of psychological constructs. And you kind of mentioned some things about where did I have it in my notes? Kind of the labeling of various tasks. And this sort of reminded me of, what you call something and what it actually measures may be 2 different things. This notion of like construct validity. What is your feeling about how we can kind of clean some of that up in order to make some of these challenges a little more tractable?
Yeah, I mean, that's exactly, you hit the nail on the head. I know you've been working in this area as well over the years, and we're sort of new to it. So I haven't tried to do the difficult task that, Russ and others have been doing, which is to try to get scientists to agree that, okay, this task measures this construct, and this one instead measures that. I haven't been involved in those discussions. And to be completely honest, the bit that I have, like in the construction of the RDoC cognitive domain, it's been frustrating because so much of it is ego driven. If you spent your 20 years of your career building up a task to study working memory, and suddenly someone tells you, well, if you look at it this way, it's not really studying that at all, it's studying this other thing. I think people are often really loath to give up their favorite task or to concede that they're not measuring a pure construct the way they thought they were. So that I think that has to sort of give way a little bit to, but what do the data tell us? The kind of thing Taylor was doing at looking at similarity across activation maps, is one way to do it. Like if they're measuring similar things, do they activate, similar brain regions? That's one way of thinking about it, right?
Sure.
So, but it's not the only way. Of course, just because regions are activated similarly doesn't mean there isn't a pattern within there that could differentiate. So as many people know, there's a lot of patterns within voxels of activation that could, again, be discriminative. So I feel like everyone, the field, we all need to kind of take a step back and say, well, is the goal to be right about what is working memory, or is the goal to come to a consensus where we can all speak a common language and move forward and be a little bit more clear in what we're saying when we use a particular terminology. So I actually do think we're getting towards that, but slowly.
It sounds like an advertisement for the use of data science to help kind of
Exactly.
sort some of this stuff out, doesn't it?
I would say that, yeah.
One of the things which is kind of interesting, and maybe you have a comment on this as well, is that a lot of these data-driven or computational topic modeling type approaches, they create a reality from what they're given.
Yeah.
And we see these biases creep in. We see this kind of particularly in machine learning and AI kind of applications for like face recognition, that there's certain ethnic groups don't get represented properly, don't get identified in the right way. And obviously, this sort of thing, you can see how this might get baked into any sort of data science application of these very rich data sets. Do you have any idea about how some of these data-driven approaches for cognition can be more robust, or at least honest about the potential biases that exist, and that might be driven by what's trendy in the literature and, may drive these things? Like you said, there's some egos involved.
Always. Yeah, this is a great question that's so important for a lot of reasons in terms of equity. And I think the best, you cannot guard against human biases because they exist on so many levels, but what you can do is build the most diverse teams. If you have programmers from all over the world and different, ethnic backgrounds, they'll be more likely to notice when these biases creep into an algorithm, and they'll be more likely to test against those kinds of things. So, having these diverse teams is critical for the science. It's not just the right thing to do.
Absolutely. One other thing is, there's other kinds of biases, though, as well. One being like publication bias. And so when you draw from a literature or from a database that is leveraging a lot of different results from around the field, you can get biases because people just publish stuff that's statistically significant. So things that aren't significant, which would greatly inform your ontology, you just don't even see them. And so again, you've created a reality out of stuff that's statistically significant. Is that helpful?
Exactly. This is the file drawer problem. What I've been heartened by is the emergence of these new journals. I think Neuroimage has one now coming out, Neuroimage Reports, and Aperture, the new OHBM journal. There's so many journals that are now realizing the need for reporting all results, good, bad, and ugly, because that's what you need in order to get a full picture of what people are actually finding. It doesn't help if only 20% of studies get published, because those are the ones people label as interesting. I'm more of a fan of publishing everything somewhere, because publishing is the way you communicate what you did. And just because what you did wasn't the most exciting thing doesn't mean that other people don't need to know that you did it.
Absolutely.
It might stop them from going down a road, or it might put them on a different path to explore, something that hasn't gotten a lot of attention. But yeah, I agree with you completely that this file drawer problem makes it hard for data science, because you can only do a meta-analysis, or you can only do a data-driven approach on data that exists or is published. So if there were, there's no studies in the brain map database that are not published, right? So Um,
Well, I could tell there's a whole sociology about data sharing in neuroscience. I was the tip of the spear of that for a little while. I could tell you horror stories, but it's just a very interesting thing. I'm pleased to see these journals taking this on and trying to do something about that. I think that's a positive step. Well, one thing I wanted to ask you about is something about the stability of these networks and how reproducible are they?
Yeah, yeah.
This is not only from a empirical point of view and a replication point of view, but also in terms of the analytic procedure which is used to identify them. Many times people use the independent components analysis method to be able to partition these networks into their different components from the data. There's some mathematical challenges, though, with some of the most widely used methods, which they sort of take a guess at to the initial state of the weighting matrix that produces the independent components analysis result. The challenge there is that while it may converge after that random guess, it may converge on a particular network structure that totally satisfies the matrix equations it's supposed to satisfy. If you run it again, you'll get a different guess and you'll get a different solution. And even if you average across all those potential solutions, it's not representative of what's actually going on. And I guess this brings to me the stability of these and whether or not they can actually be truly called functional networks if they vary so greatly, not only within a person, but between people. And you get the idea.
Yeah, it's just such a great question. It's so important. I would say the bigger question here is almost it's the same question of how many networks are there. So, and that of course any kind of data-driven clustering solution, you can either determine the number of clusters. You can say, I want 20 independent components. I want 100 independent components. You can ask the algorithm to automatically estimate the number of clusters. So it's a clustering problem as much as anything else, right? So it's not unique to brain networks or brain regions that user-defined clusters could be one approach, or data-driven clusters could be another approach. And then you have to evaluate the stability of the clusters, and you have some metric for doing that, and you might get a different number based on which metric you use. So I think these are problems that are sort of not unique to the question here we have about networks. Clustering and how many there are, what's the best solution? That's a problem generally, right? For anyone doing anything like this.
Of course, yeah.
So then it's like, which metric do you trust enough? What's amazing to me is the consistency of networks, independent of whether you do, a resting state kind of breakdown or a task meta-analysis. Or what's surprising to me is that at a given level, let's say I want 10 parcels or 10 networks, at a given resolution, there seems to be a good amount of consistency between different approaches and different data sets and different techniques. And of course, they're not identical from one to the next, but at a given resolution, it's heartening to see a good amount of stability, as it were. For example, there's almost nothing you could do. There's almost no ICA you could run that wouldn't give you some kind of default mode network.
Yeah.
Which is heartening. It means, something is there. It can break down. Certainly it can be subdivided, but, seems like there's something about the brain that lets it settle into this state. So, there's hope is what I'd like to suggest.
Yeah, it's some of our participants in our data science innovation lab are asking about the stability and changes in these networks over the lifespan. And I know that you'd mentioned some of the work of Michael Gracious, and he had done some work kind of a long time ago now, but looking at the variation between people who have Alzheimer's disease in contrast to those who are otherwise healthy agers in the context of their default mode network and changes thereof. Do you see that this is also something we should be looking at, as the changes in these networks as they alter their structure over time?
100%. And, we do lifespan network development in my lab is one of the things we're interested in. And all of the parcellations I showed you in the last few slides are done on adult subjects. And the assumption is, well, that's how the brain is parcellated. But of course, anyone who does developmental work knows that you have changes going from infancy to childhood to adolescence to adulthood in the spatial topography and the connectivity within and between networks. These are processes that develop and, again, change with aging and with disease. So of course, this is critical to understand. There have been, as far as I know, no attempts yet to parse the brain in these, in these ways across the lifespan in order to say, well, does this parcel sort of change from childhood to adulthood at the whole brain level? I think people have done this at like individual brain regions. And I think I know a group in Western Ontario who is working on this. We're looking at age bins, let's say, for example, 5 to 8 and 8 to 12 and 12 to 15, and looking at how whole brain organization in terms of the parcellation changes in those in a stepwise manner. There's other ways of doing it. And I think the availability of large data sets to do this kind of analysis is really what we're waiting on. I think there are some data sets, but not at the level that there are for HCP and the other like normal adult samples. So as soon as we get more developmental samples across the lifespan, this will be super important to address.
How can we integrate functional networks with other means for measuring networks? For example, diffusion tensor imaging, where you have a measure of structure that could be used to guide and maybe inform the functional networks that are being extracted.
Yeah, I mean, the multimodal integration part is something that is so tricky, because on the one hand, you want to say, well, okay, these two approaches, let's say structure and function, converge. But on the other hand, why should they converge? They're measuring completely different things, axons versus statistical relationships between fluctuations of both signals. So when they do converge, you say, great, these are telling us the same thing. When they don't, there's a million reasons why they couldn't. And one thing we found early on was that structure and function, like let's say diffusion metrics and functional connectivity, are seem to be very correlated in the adult brain. But when you look at children, like let's say 7 to 12, there's non-significant correlation between structural and functional connectivity. This is something we noticed a while ago. And so something is happening, something's developing. What comes first, structure or function? So you get into, all of these chicken and egg questions. Not to say we don't care about this, but we haven't made progress in this, or our lab hasn't made progress in the multimodal integration domain, but I think others have. Vince Calhoun's group and others have really thought about this more seriously.
Do you think there's any sort of opportunity for people who are developing more, complete models of brain networks that are more than just correlative, that this is correlated with that? It's rather, it's a solution to a physical network where you have, a source and you have another source, and there's a wire between them, and you could model the properties of the wire and the signal that travels over that. Are there any opportunities for those people in this area?
Yes. In fact, I before this, the last international trip I took was a conference in Nepal that was a computational neuroscience conference with, Victor Yerza and Michael Breakspear and all these great people. And my plan for sabbatical was to go visit Michael Breakspear in Australia and then visit Gustavo Deco in Barcelona. That was my original plan. Obviously, the reason for that was because of just what you said. I think computational modeling is the only way you can do this linking of different levels and really testing what parameters affect what, what outputs. And so my whole plan was to spend my year, instead of in San Diego, to spend it with those labs in those nice locations. And I'm hoping that the work that those groups are doing are going to feed into, these questions of how many networks are there and how do they change over time, because those are the only ways in which you have complete control over all the different variables. And unfortunately, I'm not doing that, as you can see. But that doesn't mean that it won't be done eventually.
International multi-hemispheric travel is going to have to be put on hold.
On hold, exactly.
A little while longer.
It's really too bad.
I think on that note, I want to thank you, Lucina, for providing us with this great overview of this very interesting area of cognitive ontologies and the role of data science in helping to identify the structure and the organization of these different cognitive constructs and what that means for the brain and for the data science that it's going to take to be able to make some traction. So thank you so much. We really appreciate chatting with you today.
Thank you. This was such a pleasure, and yeah, good luck to everyone.
Excellent. And I want to thank everybody who joined us, and we'll look forward to seeing you next Friday for the next edition of our Data Science Innovation Lab seminar series. So thank you, everybody. Have a great weekend.